So I'm going to be talking about single neuron dynamics today. And I've affectionately subtitled this A Tale of Two Neurons. I'm going to be speaking about dopamine neurons and about CA1 place cells, pyramidal cells. And what these neurons have in common is their activity. The activity of both of these neurons is tightly linked to behavior. And I'm going to be speaking about the responses of these neurons to depolarization, and specifically the role of long-term inactivation of the sodium channel. It's not slow inactivation because it happens, they inactivate quickly, but they remain inactivated for a while, is why we call it long-term. And this is work in collaboration with two other labs, with Sonia Gasparini at my institution, funded by the National Institute of Mental Health, and with Jochen Roper at Goethe University in Frankfurt, funded by NIDA. And these are the people that actually did the work. As I mentioned, Jochen Roper is PI on the grant on dopamine, and Sonia Gasparini is also a PI on the work on the hippocampus. Nicholas Hammer and Tabea Zuzu, and I see Tabea's on the call, did the experimental work in the dopamine neurons, and Crescent Cone did the experiments on hippocampus, and then Chris and Carol in my lab did the modeling. Chris Knowlton and Carol Upchurch. So I'm going to talk about the dopamine neurons first, and this is the obligatory Schultz slide. I think most of you probably know that the firing of dopamine neurons is correlated with reward or reward prediction, that they fire to a population burst in response to a unexpected reward. They can learn that a CS predicts a reward and transfer their firing back. And then they, if they expect a reward and you omit it, they pause their firing. And so dopamine neurons are pacemakers, so they don't actually need synaptic input to fire. They have a slow spontaneous firing rate, probably so that they can signal both increases and decreases in firing for better or worse than expected rewards. And in vivo spontaneous single spike firing occurs at 1 to 8 hertz. This is in vitro from my colleague Paul Shepard's lab. This is in slice, and they're just little pacemakers, rather regular. It's been known for quite some time, this is from Richards in the Katai lab, that when you depolarize these neurons from pacemaking and you cause them to fire faster, you can't cause them to fire really very fast. They once you depolarize them to the point where they're firing at 10 hertz or so, they go into depolarization block. But now, initially, experimentalists seem to have focused on the lateral part of the VTA and the substantia nigra. And they identified these characteristics of this lateral conventional population. But more recently, Stefan Lamel in Jochen Roper's lab and others have shown that there's diversity in the dopamine neurons, that they're not just a homogeneous population, but rather their subpopulations exist that can be defined by their projection areas. And so, like I said, early work focused more on lateral, and more recent work has identified neurons that are more medial, that have a different electrophysiological phenotype. And so they're studied used by injecting retrobeads. And I'm not an experimentalist, but they study them by injecting retrobeads into the projection area, and then they can see which subpopulations they're studying. And so initially, Tony Grace and others showed that the conventional population, and these action potentials are truncated, but they fire slowly. They have a prominent after hyperpolarization. And also they have a sag current. So this is the response here. We're showing a response to a pulse of hyperpolarization, and the response decays, and that's because IH is turning on. So that's a sag current. It was used often as a typical characteristic of dopamine neurons. But now that they identified these other populations and confirming that they're positive for tyrosine hydroxylase and that they are dopamine neurons, but they project to different areas, this second population, the atypical population, fires more rapidly at baseline. They don't have a prominent after hyperpolarizing potential, so there's less SK channels. And their action potentials are shorter and wider than the action potentials in dopamine neurons, as shown here. And that's reflected in the phase plane. When you plot the voltage versus the rate of change of membrane potential, you get bigger excursions in the conventional population. And from this, we infer that there are smaller spiking conductances in the atypical population. And the absence of the sag here, there's no sag in these atypicals in their response to a hyperpolarizing pulse. We infer that that means there's little to no H current. And then also when they recover from hyperpolarization, the atypical population has this ramp-like response, and there's a delay until firing is resumed. And that has been shown to be due to slower inactivation of the A-type potassium current in these neurons. So given that there's these differences in their electrophysiology, what does that mean functionally? And Stefan Lamel applied triangular current ramps to these. The ramps aren't shown, but you can infer them from the time course of the membrane potential. What's shown on the left is his retrobead and TH data showing these are dopamine neurons and that they project to these different areas. As I said earlier, the conventional ones were more the ones that project to the striatum, the dorsal striatum, and also that project to the lateral shell of the nucleus accumbens. So these were the typical dopamines that were initially identified. Then the newer identified atypical population responds differently to triangular pulses of current. So they're trying to simulate what happens during a burst. So dopamine neurons fire from 1 to 8 hertz at baseline, but then if per Tony Grace's definition, when they fire faster than 8 hertz, they call that a burst. And as we've seen, bursts do have functional significance in these cells. The conventional cells, as I stated earlier, only can fire at about 10 hertz max. These atypical ones can go up to 20 hertz. And the atypical ones project to cortex, amygdala, the core of the nucleus accumbens, and the medial shell of the nucleus accumbens. And so you can see for a weak ramp on the left-hand side, they do not go into depolarization block, but for a stronger current ramp, these neurons are forced into depolarization block. So they're depolarized, but they're not firing because there's no availability of sodium channels. And they also go into depolarization block in a qualitatively different way. You can see that the atypical population stops firing, but the action potentials taper off and become very small. Whereas in the conventional population, you have an abrupt failure of action potentials. And it's also very important to note that the maximum rate is much higher. The maximum rate prior to entering depolarization block is higher in the atypical population. All right, so Chris Knowlton in my lab, and this work was recently published in PLOS Computational Biology, created a single compartment model to capture the essence of the responses of these neurons to the triangular pulses. It consists of an equivalent circuit with the currents that are known to exist in these cells, a sodium channel, a leak, delayed rectifier, an A-type, the SK, and several calcium channels, and also the H current. And he also performed a material balance on calcium in order to have a pool to activate the calcium-activated SK potassium channel. So I mentioned that I'm talking about cumulative inactivation of, in this case, the NAV 1.2 sodium channel. And this is work from Ding et al. In 2011, where they take nucleated patches and they apply brief pulses of voltage clamp to 0 millivolts. And if you apply them close enough together, and that's 100 milliseconds, the sodium current that you evoke decreases over time. And so in order to capture this aspect of the model, Chris took a model from the literature from Jeanette Kotelewski's lab that was it was initially a 6-state model. He dropped it down to a 5-state model. And the rate transitions between all these Markov states are voltage dependent. Voltage dependence is given here. There's a maximum rate, and then a Boltzmann dependence. And in some cases, it's a sum of a Boltzmann. But he adjusts, and I emphasize this I1. So we have an I1 state, which is the fast inactivated state corresponding to H in the M cubed H Hodgkin-Huxley formulation. We've got an open state, and of course, current only flows when the model is in the open state. We have two closed states, and as I said, an inactivated state, and then another inactivated state that's more long-term and more difficult to get out of. And the parameter that we're going to be talking about is the rate of the transition rate from the short-term inactivated to the long-term inactivated state. And that's going to be a critical parameter in the model. So Chris, I showed you before the data from Stefan Lammel's paper on the atypical population and their spontaneous pacemaking rate, their action potential shape, and the shape of the action potential in the phase plane of voltage versus the derivative of membrane potential, and also their responses to hyperpolarization. And Chris calibrated the parameters of the single compartment equivalent circuit model that I showed you to qualitatively capture the features of this atypical population. But now, if he ignored the long-term inactivated state, and so this is the data on the left-hand side for a weak triangular current ramp and a strong triangular current ramp. You enter depolarization block here. Now, if he gives the model triangular current ramps, but neglects to include the long-term inactivated state, then the responses are not qualitatively similar to the experiment at all. For the weak pulses, you don't go into depolarization block. For a strong pulse, you do achieve depolarization block, but on the downstroke, the channels recover from inactivation and resume spiking, totally in contradiction with the experimental data. So in order to better fit the data, Chris implemented this long-term inactivated state. As I said, the key parameter is this transition rate between I1 and I2, the transition into long-term inactivation. And so what was happening before, again, here's the membrane potential of the model for the weak ramps and the strong ramps. The blue is occupancy in the short-term inactivated state. So in the weak ramps, you don't drive the neuron into depolarization block. You still have some availability of your sodium current. You know, if your inactivation, if your occupancy in the inactivated state is 1, that means all the channels are in that state and you don't have anything that's available to open immediately. In depolarization block, all of the channels went into this short-term inactivated state, but as soon as you start decreasing the applied current and hyperpolarize and the neurons becoming less depolarized, then channels fell out of that state and you were able to resume spiking. However, if you incorporate long-term inactivation, and now the color codes are red is for all inactivated states, blue is still for the fast inactivated state, and green is for the long-term inactivated state. What happens now is much more similar to what you see in the data. You get, with each spike, you get accumulation of occupancy in the long-term inactivated state, and that then reduces the availability of channels to go into any of the other states, the closed and open state, for example. And you get adaptation, you get slow firing at the end of the pulse. And as for a strong enough pulse, now you get so much cumulative inactivation of the sodium channel that you don't, and here the red, basically all of your channels are in one or the other inactivated state. So you go into depolarization block. You're depolarized, but you don't have enough availability of sodium channels to keep spiking. So you go into depolarization block. And you'll note that the mode of entry into depolarization block is qualitatively similar. You get fast firing and you get a gradual decrease in the amplitude of the action potentials. All right, so my experimental collaborator, Jochen Roper, wanted Chris to double check and make sure that none of the known manipulations, none of the known differences between the populations accounted for the difference in maximum rate or for the different mode of entry into depolarization block. And so what we show on the left-hand side is just, this is the control panel from the previous slide where we're driving the atypical model into depolarization block. And as we said, we know these cells have little sag current. That doesn't make any difference. They recover much more slowly from the A-type channel recovers much more slowly from inactivation, and they have less SK current, and they have, we inferred that they have stronger spiking conductances, but none of these manipulations slow down the maximum rate. They fire just as fast, if not faster, and it doesn't change qualitatively the mode of entry in the depolarization block. All right, so this is actually the key insight from the paper. So this is, once again, this is the atypical model with the amount of long-term, the entry into the long-term inactivated state that Chris calibrated to match the data. And the adaptation that you see, and the entry into depolarization block. But if he changes one parameter, and that is the I1 to I2 transition, the transition between the short-term inactivated and the long-term inactivated state, that's the only parameter difference between the top traces and the bottom traces. And you can see that dramatically slows the maximum rate that's observed before you go into depolarization block. And it also dramatically changes qualitatively the mode of entry into depolarization block. And so again, you can see accumulation of long-term inactivation, but you see each spike makes a very small bump in the long-term inactivation for the standard parameter set for atypical. But when he increases the transition into I2, from I1 to I2, now you get much more inactivation with each spike, much more long-term inactivation with each spike, until you go into depolarization block. And the reason they're qualitatively different is the bifurcation diagrams on the right-hand side show that for the atypical with the standard parameters, there's a supercritical hop bifurcation. So the black is the envelope of the oscillation, the maximum and the minimum membrane potential. And you can see it just gradually, as in a supercritical hop, the amplitude gradually decays until you stabilize the fixed point. And this is an unstable fixed point here. What happens when you increase the tendency to go into the long-term inactivated state, you have less available sodium current, and you stabilize this fixed point such that you get a subcritical hop bifurcation. But as you increase the current, you spike at a fairly constant amplitude until you fall into the saddle node of periodics, where the unstable limit cycle collides with the stable limit cycle, and spiking abruptly stops and you go to the fixed point. So this is the key insight, is that changing this parameter changes the bifurcation structure of the model, and therefore the rate, the maximum rate that you can have before you go into depolarization block, and also the mode, whether it's gradual or abrupt, of entry into depolarization block. All right, so I've showed this slide before. This is the calibration of, well, the basis for calibration of the rate of entry into long-term inactivation. So this is the sodium current recorded in a nucleated patches, as I stated before, in response to repeated voltage clamp steps. And so Chris used that data to calibrate the conventional model. So we've been talking up to now exclusively about the model of the atypical population. We're now going to transition and look at the conventional population. And so what the calibration that Chris did for long-term inactivation in the atypical cells, that's never actually been measured. So that's a prediction. We're predicting what is long-term, what is the rate of entry into long-term inactivation in the atypical population going to look like? You know, how much is it going to decrease with each repeated voltage clamp pulse? But for the conventional model, the data was out there. And so Chris calibrated the model according to the data, and also made parameter changes based on all what I've gone over several times now, the known differences between the two populations. We know they have different IH. We know that KV4 recovers more slowly, or goes into inactivation and recovers more slowly from inactivation in the atypical population. And we surmise that the spiking conductances are stronger based on data, and that the SK is also stronger in the conventional population because of more prominent HP. But then we also postulate that there's a this is a prediction that there's another difference that had not been uncovered before, and that is that you have a larger tendency, a higher max rate to go into the long-term inactivated state in the conventional population. And so this is a busy slide, but it's a summary slide, and it's showing that previously we showed that the calibrated model is qualitatively the same to the data. So model on the left, data on the right. We've already seen that the atypical was calibrated, but now the conventional model is also calibrated per the data from the Lamel paper. And recall again, these action potentials are truncated, but we have a slower pacemaking rate, a deeper after hyperpolarizing potential, taller, thinner spikes, and larger excursions in the phase plane that shows the dVdt, the derivatives during the upstroke and the downstroke. And we also have a faster rebound from hyperpolarization due to the presence of the IH sag current and due to faster recovery from a faster inactivation of the A-type channel. So basically, this is a calibration slide that shows that we captured it. Now that we've calibrated the model, is it able to capture the responses to depolarizing bumps? And on the right-hand side, again, is the data from Lamel. And we've already shown many times that the model is able to replicate the responses to both weak and strong current pulses. But then we also show now the calibrated conventional model exhibits adaptation to the triangular ramp for the weak ones and goes into depolarization block abruptly and at low maximum firing frequencies. So we're able to capture the essential differences between these subpopulations. Then Chris went on to make one final prediction that was tested in the Roper lab. And so this is basically, okay, so this shows pacemaking. And during pacemaking, this is the occupancy here. So green is the slowly inactivated state. Blue is the short-term inactivated state. And I didn't mean to say slow, I mean to say long-term, because you enter it quickly, you just come out of it slowly. This is total occupancy in both inactivated states. And then this is everything else, the available pool, O, C1, and C2. And the key to maintaining pacemaking is that during a spike, you inactivate channels. During the interspike interval, and in particular during the AHP, you recover from inactivation. And so the amount of recovery has to be exactly equal to the amount of inactivation that was induced by a spike. And if your interspike interval becomes too short that you can no longer recover, then you go into depolarization block. And that's what's happened. That's what happens here when you give it a step current. So these are steps. This is not a ramp, but we're going to give it a step current, go into depolarization block, and then do a test pulse to determine whether you can evoke a spike while you're in depolarization block. And on the bottom here, then this is the rate of change of the membrane potential, which is a readout. It can, of how much sodium you have available. So this maximum dVdt that you reach on the upstroke, that's a readout of sodium availability. And you have to go to a small scale to see the difference. But because of the method of failure and the fact that the atypicals keep spiking until there's nothing, no gas left in the tank, whereas the conventionals stop at that saddle node of periodics, you have to look on a very small scale, but there's almost no availability of sodium channels once you go into depolarization block for the atypical, but you've got a small reserve. You've got a few percent reserve of available channels in the conventional population. So if you give them an additional depolarizing pulse when they're already in depolarization block, they're still capable of firing an action potential. So this might have some significance for signal processing in dopamine neurons. So the Roper lab tested this on, they took, Tabea, who's in the audience, looked at neurons that projected to the nucleus accumbens medial shell, which are representative of the atypical population, and neurons that project to the lateral shell, which are representative of the conventional population, and tested exactly Chris's protocol. They put them into depolarization block and then gave them an extra bump of depolarizing current. And sure enough, and then again, here's the DVT that is a readout of availability of the sodium channels. And sure enough, they were able to evoke an action potential much more often than not in the conventional population, but not in the atypical population. And they again showed their peak burst frequency is higher in the atypical than in the conventional. The evoked amplitude of this spike was much higher in the conventional population. The max DVDT is also much higher in the conventional population. So this is a success story of a prediction that was validated. So this is the end of the part of the talk on dopamine neurons. I'm going to transition to CA1 pyramidal neurons. I just want to reiterate that conventional cells go into depolarization block via saddle node periodic bifurcation, the atypical via supercritical hop. This accounts for the main differences in their modes of entry into depolarization block, and the model prediction of being able to spike even though you're in depolarization block is true for conventional, but not to atypical cells. So I'm going to transition to place cells, pyramidal cells. They're known to, again, the link of these cells to behavior is that they're known to fire preferentially when on a linear track, a rodent is in a particular field of the environment, but they encode sequences, and probably this is generalized to any kind of episodic memory. But in any case, we'll focus on place cell firing. Now, it is assumed that they receive a bump in their synaptic input when they're in their preferred place, and in fact, in awake head-fixed mice, this has actually been recorded that there's a bump in the depolarizing envelope, and the firing rate actually peaks in the center of the place field. That's how the place field is defined. But the depolarizing bump actually peaks towards the end of the place field. So that's this is a repeat. So this is firing rate as a function of position in the place field. And now the envelope in the membrane potential synaptic bump that they're receiving when they're in there. And these are in a virtual environment, so they're in their virtual place field. And this is explained by a paper in anesthetized rodents where they injected a bump of current, and they find that it fires much more on the up ramp than on the down ramp. So there's some sort of adaptation mechanism operative here, just as there was in the dopamine neuron. In the dopamine neuron. So there's adaptation, and that at the same level of current on the down ramp, they fire less than on the up ramp. And so Crescent Combe in Sonia Gasparini's lab recorded from CA1 neurons, and all the dopamine neuron work, all the recordings and all the current injection was done at the soma. But in CA1, they have the capacity of injecting and recording in the soma, or in injecting and recording at different points along the apical dendrites. And they're going to, and let me go back one. And so they inject triangular ramps of current, just as was done in the Lamel paper. But now we're going to inject them into CA1 neurons. Okay, so the experimental ramp responses are shown here in Upchurch et al. Submitted. They give, this is a brief 2-second depolarizing ramp. This is a longer ramp of 10 milliseconds. Could maybe make an analogy to different running speeds through the place field. And what's plotted here is the 1 over the ISI, the interspike interval, the instantaneous frequency. And what you note here is that you have fewer spikes on the down ramp, and those the spikes on the down ramp have larger ISIs or slower frequencies. So you have more spikes, faster frequencies on the up ramp, fewer spikes, lower frequencies on the down ramp. That's what's quantified here. It's hysteresis. The effect is more prominent in the dendrites, shown at the bottom. You have somewhat more adaptation in the dendrites. And you can also see that there's a decrease in amplitude of spike height in the dendrites. This is a summary of the data, and just showing that if you take the ratio of spikes on the up ramp minus spikes on the down ramp, and divide it by all the spikes, then a ratio of 1 will give you perfect adaptation. You only have spikes on the up ramp. A ratio of -1 would mean you only had spikes on the down ramp. Here we've got all positive ratios, and you can see that the adaptation ratio is larger in the dendrite, at least for the short ramps. All right, so Carol Upchurch in my lab took an existing multi-compartmental and now we're not going to use a single compartment because we're injecting at different points on the neuron. We're going to use a multi-compartmental model of a CA1 pyramidal neuron. It is, again, an equivalent circuit model that is in each compartment, but also the parameters are spatially inhomogeneous. Carol took the original model, the model that we had published previously, Combe et al., and added a second delayed rectifier, KV2, in addition to KV1. And she implemented the Markov model in the soma and apical dendrites of the CA1 neuron. And this is just a calibration of the passive properties to show that the ballpark response of the neuron to a 200 picoamp, 500 millisecond hyperpolarizing pulse is ballparked. So this is the calibration data. I need to make a note based on this work is under review and based on a suggestion from the reviewer. States C1 and C2, closed 1 and closed 2 from the other model were merged into a single closed state. And this did not actually affect our results. So this is a difference. Again, we're going to be manipulating the I1 to I2 transition. And the reason some arrows are thick and some are small is these are very small rates compared to the rate transition in the other direction. So they're indicated small. But again, now I1, I2, the transition tendency to enter into the long-term inactivated state has been shown to be a function of distance from the soma along the apical dendrite. This is data from the Spruston lab. And so again, this is now cell-attached patches with voltage clamp steps that are repeatedly applied. And you can see there's a little adaptation. I mean, a little decrease at the soma, but much more prominent decrease in the current evoked as you get farther from the soma. So Carol adjusted the I1, I2 to be as a function of distance from the soma in order to replicate the percent steady state inactivation that you get after applying a train of pulses, and also to replicate the time course of entry into the slow, they called it slow cumulative sodium channel inactivation. I call it long-term inactivation. All right, so with this calibrated model, she's able to capture the essential features of the shape of the spiking in response to a depolarizing ramp. You get more spikes on the up ramp than the down ramp, and you get slower spikes, slower frequencies on the down ramp. So the closed circles are the up ramp, open circles are the down ramp. And she gets similar results to what was observed in the soma, and you get more prominent adaptation in the dendrites, and you also get much more prominent decrease of the amplitude in the dendrites. So we're going to go to the Markov model for an explanation of these phenomena. Again, what you get is, okay, so green is the I2 state, blue is again the I1 state, open is orange, and the closed is this light blue. And what you see is that you again get accumulation of channels in the long-term inactivated state, and that then robs from the from I1 and a closed and open from the other states because they're sequestered in this long-term inactivation state and not available. So that causes both the decrease in amplitude and the change in frequency. But what we noticed is that this struck me as very unusual. During the dendritic ramps, the amplitude goes down, but then it starts to recover. And so I initially postulated that there had to be another mechanism, a slow potassium current that was causing the adaptation to continue to get, the frequency continues to get slower while the amplitude of the action potential is increasing. And what the unusual thing is that Carol captured this behavior in the model, the slowing of the frequency while the amplitude is growing without even trying. It just fell out of the model. And so you can see that in the short ramps, accumulation of inactivation occurs up until the end of spiking. However, in the longer ramps, as the amount of depolarization is decreased and the cell's starting to hyperpolarize somewhat, you get recovery. You have channels that are leaving the long-term inactivated state, and that makes more channels available to go into the other states. So it was puzzling to me how this I2 could be responsible both for the decrease in spike height and the change in frequency, slowing in frequency, but that it differentially regulated these two quantities. And so the answer is, in brief, that the amplitude is regulated strictly by occupancy in the closed state. Those are the only ones that are immediately available to open just prior. So if you look just prior to a spike and orienting you, this is the up ramp. So we're moving in this direction. And what the axis here is current, injected current. So I plotted it in these coordinates in order to show hysteresis. So the up ramp, that's the amount of closed channels that are available to open just prior to the first spike. Then as you go along, occupancy of C is decreasing, but then on the down ramp, occupancy in C starts to increase a bit due to the fact that you're hyperpolarizing, and so this is and the arrows show what is the amount of C2 that's available just prior to a spike. Now you have hysteresis in I1 as well. So on the up ramp, it's increasing, and on the down ramp, it's decreasing because of the hyperpolarizing influence, but it's still way above what it was on the down ramp. So you have more channels in the I2 state on the down ramp, and that's causing these effects. But the effect on spike height is caused strictly by the occupancy in C. And so you can plot occupancy in C just prior to a spike and spike height measured from threshold to the peak. And it tightly correlates with occupancy in C. So the spike height can recover because occupancy in C is recovering. It's coming up. It's not at the levels that it was on the up ramp, but nonetheless, it's recovering, so your amplitude can recover. Your frequency does not recover. You're still getting slower. And that's because this O state lags behind the C state in its recovery. And again, I'm showing the hysteresis in the I2 state that's driving everything. So up ramp, down ramp, and this is occupancy in O. Occupancy in O is tiny. You can see this is to the 4th power, but it's significant because it translates to currents on the order of 100 picoamps. And of course, you have much less current on the down ramp as compared to the up ramp, and that accounts for the difference in frequency. And you're not getting any recovery in this O state on the down ramp. It's pretty flat here. Let me get my pointer going. It's pretty flat. So therefore, you're still slowing. Your current you're way below the amount of current that you were, and below here, minus 80 picoamps is less than minus 125 picoamps, because here we're talking with the negative axis. So this is less current. You got less current. You have much slower spiking. And just Carol plotted the net current, summed it over all compartments of the model. So this is the net persistent sodium current. It's a few hundred it's 100 picoamps, say, much smaller than the large currents, nanoamp currents that you get during the spike transiently, but still very significant enough to influence the frequency. And she plotted the total current, which is, you can see it's pretty flat here. So the total current basically goes to 0 when your membrane potential is flat. So two more points to make. Model predictions. Model prediction that decreasing long-term inactivation is going to decrease adaptation. So in the model, we can just take I2 out, and there's no more hysteresis. The spikes on the up ramp, you have the same number, and they're at the same frequency than on the down ramp. This is for both the soma and the dendrite. Then in experiments, you can use PKC. Well, you can use a formyl ester, PDBU, to activate PKC, PKC phosphorylates the sodium channel, and that has been shown to decrease long-term inactivation. So as predicted, putting on the 4-bol ester decreases adaptation, both in the soma and at the dendrite. And this is summary data showing that for matched pairs, the adaptation ratio goes down. And conversely, when you increase adaptation, there is an endogenous factor that binds to a site on the sodium channel, and that causes long-term inactivation. If you don't have that endogenous factor, like for example, in fast spiking interneurons, you have the same sodium channel, but you don't get long-term inactivation. But so Sonia Gasparini's lab applied this peptide, and there's not a real control because the peptide is inside the patch clamp pipette. But as you put this peptide in, you can cause great adaptation in the soma here. So this is an example with no peptide in the soma. This is an example with a high concentration of peptide in the soma, and you can make it adapt as much as a dendrite. You get just here's one spike on the down ramp. In the model, if you increase the rate of entry into the state I2, then you can get rid of you can make the adaptation much stronger here. You get very few spikes. And this is just summary data showing the normalized adaptation ratio with no peptide, and as you increase the concentration of peptide. So to summarize, I2, the entry into this long-term inactivated state, controls decreasing dendritic spike height via controlling the available pool. It increases spike frequency adaptation. It controls the frequency by decreasing availability in the open pool. And then also so that's the main conclusion, and also the fact that we can replicate the difference in the dendrites and the soma, and the model predictions were borne out. And adaptation due to this long-term inactivation can affect place field centers of mass in vivo. Then one final quick point is we went back to the dopamine neuron model, and we had not noticed that in the dopamine model, you also get a decrease in amplitude without a recovery, and then you get some recovery in amplitude, but your frequency keeps getting slower, no recovery in the frequency. And so Chris went back and he plotted C1 plus C2, so basically all the closed states, and he found that the same thing happens, that you get some recovery in this closed state, and that allows your spike height to recovery, but you don't get recovery in O, which I'm not plotting. So the frequency continues to slow while the amplitude increases. And my overall conclusions are that in two disparate neuronal populations with two different NAV channel subtypes, I didn't emphasize that, but one of them has NAV 1.2 and the other has 1.6. It limits the firing rate in response to sustained depolarization. So how general is this mechanism? And finally, just a thought question. We're talking to now Uchita about how multiple scales of adaptation mechanisms could endow a neuron with fractional differentiation to represent a compromise between the input signal and its derivative.